So in order to visualize this data in three-dimensional space, what we're going to want to do is come up here to this top menu and click on the scatter plot. I'm going to do that right now. This will bring up this window. Um, so this is a principal component analysis plot. So in order to visualize this data in three-dimensional space, what we're going to want to do is come up to the menu and click on scatter plot. This will kick us to a new window. Again, this is a principal component analysis plot, or PCA plot. Without digging into the mathematics and talking about eigenvectors and matrix theory, what you should understand about a PCA plot is it uses all the variables, or at least the variables that change within a group of samples. In this case, this is our biosets, and plots them based on these variables in three-dimensional space, so that samples or biosets that are more like each other will be closer together, while biosets or samples that are more dissimilar will be farther apart. And this just kind of gives us a good way to get an overview of the data. We can actually spin this data any way we want. We go over here to click on this kind of curve looking arrow, and now we can kind of manipulate this. Right now I have this colored based on the 8 different omic studies, gene expression studies that we used in our meta-analysis. And you can see kind of, obviously some of them probably use the same samples. We can see these green ones kind of grouping here. But for the most part, they're pretty dispersed, and I don't see all these samples in the same area. So I don't think there's a huge batch effect. What I think is that the variables in our samples, like the type 1 versus type 2, I think this is what's driving this diversity that we're seeing. But we can color this any way we want. I can go over here, I'll click on my pointer. We can color this based on the type of cancer that we tested. I'll move this over here. Again, you can see low grade, so type 1 would be the less severe form of papillary renal cell carcinoma. We have low grade. We have these non-specific forms, so kind of a combination. Don't know if it's type 1 or type 2. And then all the purple ones are type 2. We can also, and you're kind of seeing the separation now. Now you're seeing those that are of similar types, like this type 1, seem to group together, even though they're from different studies. You can see some of these other, and then type 2, you're kind of seeing this lower plane here, kind of differentiated in itself. We can now basically shape by the control tissue that we used. So we can see down here that the sporadic and type 1, and the excellent survival, these are all over here. These were cancer versus cancer, and you can see they're very different. So everything over here was cancer compared to either normal kidney or normal adjacent kidney. You see it kind of differentiating itself as well. I think what you're going to see is, and what's nice about this, is when we do the clustering in the next video, what we'll do is you'll see how these samples not only are related, but now we can pick out genes that we feel kind of differentiate these biosets from each other. So that's actually what we're going to do in the next video. If I go down here, this is what we're going to produce, is a cluster, a hierarchical clustering in the form of a heat map, and that way we can see all our different samples on our columns and kind of start looking at some of these different clusters of genes that differentiate these different types. And by looking at these clusters, we're going to learn a lot about this cancer. So this concludes section 3 of our meta-analysis series. In our next videos, like I said, what we're going to do is now visualize the data using clustering and heat maps. And what we'll do is then take some of these groups that we feel differentiate the different comparisons that we looked at and start looking at the biology behind that. So in the next series of videos, what we're going to do is start digging into the biology of this. What makes this cancer, and what differentiates, say, the poor versus excellent survival. Until next time, Thank you very much. This is Michael Edwards.